Today's date from the Daily Mail: the human brains being grown outside the body, lab making miniature organs in a jar, is revealed. In 2013, scientists used stem cells to grow 3D tissue that mimics a brain. Now researchers over the world are working on making these mini brains. Skin cells are transformed into stem cells, which are grown into brain cells. The brains are being used for researching disorders unique to humans. From what makes us right or left-handed to why we develop autism, there are many mysteries about the human brain we are yet to solve. Some of these questions can be answered by studying the brain or brains of other animals like mice, for example. But this isn't possible for other phenomena that are unique to human brains. Researchers are now growing hundreds of tiny human brains in labs in an attempt to understand what gives us unique disorders like autism and schizophrenia, and the method they use to create these brains is surprisingly simple. Researchers are now growing hundreds of tiny human brains in labs. A magnified picture of an organoid 3 to 4 millimeters across with a structure similar to that of a human brain is shown. Scientists across the world are developing cerebral organoids, or mini brains, to solve a variety of problems. Many of these groups are trying to understand other complex neurological diseases that are unique to humans, like autism and schizophrenia. One such researcher is Madeline Lancaster, who works at the Medical Research Council Laboratory of Molecular Medicine in Cambridge. The brains are created using cells. The team uses skin cells, but they could start with any cell type. The brains develop in the same way you would see in an embryo, Dr. Lancaster told BBC Future. They turn these cells into stem cells using proteins, and as they grow, brain cells begin to develop. The researchers starved the cells, and for an unknown reason, the brain cells seem to be the most robust ones, so they survive. These brain cells are placed in a special jelly and put into an incubator. The researchers in Dr. Lancaster's lab are using these brains to study a variety of conditions. Our current interests focus are on other neurodevelopment disorders like autism and intellectual disability by introducing mutations seen in these disorders and examining their roles in pathogenesis in the context of organoid development, Dr. Lancaster says on her project page. Scientists created pea-sized brains from a patient's skin that could lead to cures for common neurological disorders. First brain in a bottle was grown by a stem cell scientist in 2013 who hoped it would lead to treatments for neurological and mental diseases. The organoids, 3 to 4 millimeters across, have a structure similar to that of an immature human brain. Just like a normal brain, the organoids are divided up into gray matter made up of neurons and white matter, a fatty tissue composed of their spinally tails. Each is also composed of a specific regions like the human brain. Professor Jürgen Noblick of the Institute of Molecular Biotechnology in Vienna derived in the iPS cells induced pluripotent stem cells from the connective tissue of a patient with microcephaly. This is a rare but devastating genetic disorder in which the brain size is dramatically reduced, leaving the sufferer with severe mental disability. Like many neurological conditions, the disease has been difficult to study in mice because they do not share the same brain complexity as us. The team used a 3D matrix scaffold that mimics the environment of a human embryo and special lab devices called spinning bioreactors, which produce nutrients and oxygen to grow the brains. Using stem cells, master cells that can turn into almost any cell type in the body, researchers grew skin cells into a 3D tissue that mimics a brain. Image shows a cross-section of an entire organoid. Ultimately, we would like to use them to study more common disorders like schizophrenia or autism, as it has been shown the underlying defects occur during the development of the brain, Professor Noblick said at the time. We are satisfied, or we hope, we will be able to model some of these defects as well. The original goal was to produce a biological tool that can be used to investigate the workings of the brain, better understand brain diseases, and test new drugs. The goal for many researchers is to develop a brain exactly like a human's, but some researchers say this would be a step too far. Dr. Martin Coeth from the Cognition Institute at the University of Plymouth questioned why anyone would ever want to create a real human brain. A human brain that was fully working would be conscious, have hopes, dreams, feel, and pain, and would ask questions about what we were doing to it, he said. Something we have grown in the lab, but on a much simpler level than a human brain, might be hooked up to electronic eyes, ears, and hands and be taught to do something, maybe something that is as sophisticated as many simple living creatures. That doesn't seem so far off to me. Isn't it amazing what man is trying to do in the laboratories? But thou, O Daniel, shut up the words and seal the book, even to the time of the end. Many shall run to and fro, and knowledge shall be increased. And knowledge shall be increased. No, the Bible doesn't have any sense in it. It's just a make-believe book that, there's no knowledge that's being increased in these last days, in the last 100 years. We're still riding in buggies like we did 100 years ago, right? Thank you for listening. God bless.